thank you. I am very, very glad to be here, and I thank the organizers for inviting me to this beautiful place. Today I will talk about our studies aimed at trying to understand the processes of islet cell type interconversion that we see in the pancreas of adult mice. So my talk today will begin with a brief overview of the experimental setup that led us to identify this phenomena of islet cell type interconversion in mice, and how these observations could lead to the development of new therapies to treat diabetes. And then today I choose to present a number of experiments that are aimed at trying to address the nature of the signals that drive this plasticity, how alpha cells can change their identity in these mice. And also I will present a few studies, ongoing studies, on human alpha cells and how we can make them to start making insulin. In both cases, these studies are unpublished, and like Richard said, like Richard explained, I will ask you please not to take photos because we want to protect the young investigators that have to publish these results. And I will prevent any diffusion of this privileged information before we haven't had the acceptance of these papers. So please, let's think about them. So before I go into the details of our work, I would like to remind you something that we already know about how cells and tissues respond to injury, to damage. Until recently, we have been told that differentiated cells remain with the same identity for the whole life, but this is not true. So what do we know about regeneration after damage? So in tissues with high renewal rate, tissues in which there is a strong capacity of proliferation, we have this response which can be called as adaptive cellular proliferation, whereby differentiated cells, sister cells, remaining cells can proliferate, like in liver, or we have recruitment of stem or progenitor cells that undergo division, like in skeletal muscle. But what happens in tissues in which there is a lot of quiescence, lazy cells that do not proliferate? So we have, and then we start to understand that what happens is what can be globally called as adaptive cellular reprogramming or cell conversion or change of identity of differentiated cells. So what is more frequent is that there is a transient reprogramming of cells that promote healing. And this is what happens, for instance, with the Schwann cells that help the regrowth of axons. But there's also another kind of response that is less well known, although it was described a long time ago. This was well characterized by the great geneticist Morgan more than a century ago. So this is what is called the reprogramming of differentiated cells. So this is the generation of replacement cells by reprogramming. And indeed, this is something that happens in frog inverts at the levels of the eye, for instance, this iris to lens cell conversion. Or in mammals, we have this hepatocyte to biliary cell conversion, or this auditory epithelium cell conversion within the cochlea, in the inner ear. And what we have seen in the pancreas of mice is that there is also this kind of phenomena of islet non-beta to beta cell conversion. So this reprogramming may happen through two different mechanisms, which are well described in the animal kingdom. One is the so-called direct conversion, whereby you have the formation of transitional cells with a hybrid phenotype. And this was called morphallaxis by Morgan. And the other process is the de-differentiation of differentiated cells that enter the cell cycle before re-differentiating again. And that process was called epimorphosis. As I will show you, we have seen these two processes to happen in the islets of mice. So the question is, we want to foster beta cell regeneration, And the idea is whether we can exploit this naturally occurring adaptive cellular plasticity to promote a cell regenerative therapy for diabetes. So let me summarize first what are the present day and the tentative strategies that are devised to try to preserve or reconstitute the beta cell mass and the beta cell function with patients. So I will not talk about the immunomodulation, the protection of beta cells against autoimmunity in type 1 diabetes. This is something obvious and interesting. But what I'm going to discuss in this slide is the two possibilities which could be called like that. The directed cell replacement approach versus the innate cell regeneration approach. So today what doctors do to cope with diabetes is to try to improve insulin secretion, to improve beta cell survival, to try to induce replication of remaining beta cells. These are beta cell regeneration strategies. When things go wrong, the only solution is transplantation, whether of isolated islets or of total pancreas. This is a replacement approach. Now, as we know, we have seen, there are many laboratories around the world that are trying to obtain ex vivo beta-like cells, whether from pluripotent cells, embryonic stem cells, or iPS cells, or even from adult differentiated cells. So this is, again, a cell replacement approach, because the ultimate goal is to achieve this autologous transplantation of cells that were obtained originally from the patient. In my perspective, this approach is powerful and extremely useful to study disease modeling and to develop drugs by screens. But I personally doubt that we will be able to use these cells to cure diabetes. This is my personal bias, and I know that many people don't share this view. Other approach, other laboratories are trying more weird solutions, which is this in vivo adult cell reprogramming, but from extra pancreatic tissues. So there are laboratories trying to make liver cells to become insulin producers, gallbladder, thymus, intestine, many other cells. This, again, is a cell replacement approach. And what we have seen in the pancreas is that there is this capacity of adaptive adult cell reprogramming, whereby non-beta cells within islets become insulin producers. This is a pancreas regeneration approach. Trying to understand this and foster this process is a regeneration approach. So it all began with this very simple question. We wanted to determine whether the adult pancreas in an adult mouse, can generate new beta cells after their total destruction, like in type 1 diabetes. And to do that, what we follow is a very simple, conceptually simple experiment, which is the combination of the in vivo cell ablation, the destruction of beta cells through the use of the stereotoxin, in which mice in which the human receptor for the stereotoxin is borne by beta cells, combined with the labeling of cells to try to follow their progeny and see whether these cells can contribute to the formation of new insulin-producing cells. That was done in transgenic mice. Let me remind you how it works. The stereotoxin is a soluble secreted toxin which has 3 subunits, A, B, T. This is made by Corynebacterium diphtheria. And the B subunit is the natural ligand to the receptor, which is this membrane form of heparin-binding EGF. So once internalized within the endophagosome, the change of the pH makes that the T domain undergoes a conformation change, and then the A domain, which is the active part of the toxin, is translocated into the cytoplasm. And once into the cytoplasm of a cell, one single molecule of the stereotoxin A is enough to catalyze the adenoribosylation of this elongation factor, and this results in the prevention of any protein synthesis, and the cell dies very quickly by a process of apoptosis. This is one islet of such transgenic mice in which you can see plenty of insulin-producing beta cells in red. And after treatment with this stereotoxin, you see an islet in which you can barely see only glucagon and somatostatin-producing cells. So the system is very efficient. And by doing this, what we observe is that if we keep the mice alive with insulin therapy, with exogenous insulin, with time we see the appearance of new insulin-producing cells. That was the first surprise, in fact, because I was convinced personally that the whole pancreas cannot regrow new insulin-producing cells. Anyway, we wanted then to determine the origin of these newly formed insulin cells, and for that, we did a number of experiments, among which we genetically pre-label the beta cells before killing them in order to try to see whether the escaping cells were the ones that were contributing to this regeneration. And the fact was that the newly formed cells, in fact, are not tagged, meaning that they were not escaping beta cells. Doing similar experiments, we could finally label genetically, irreversibly, the alpha cells in a mouse with a pulse and chase approach, and then destroy the beta cells. And what we could observe after a period of at least a month of regeneration was that the newly formed insulin-producing cells were tagged, meaning that they were, in fact, reprogrammed alpha cells. So how can we label alpha cells in a mouse in a conditional way, in a pulse and chase approach? We may have this setup in which these transgenes are present allow us to label alpha cells by providing doxycycline, which is a tetracycline analog, in drinking water. In this situation, alpha cells can activate the expression of Cre recombinase, which in turn catalyzes the excision of a stop termination sequence in a reporter transgene. And in this case, we have expression of a reporter molecule. In this example is yellow fluorescent protein, YFP. And in this way, by stopping doxycycline treatment, we can pulse label alpha cells in an adult mouse. So after that, we can decide to destroy the beta cells with this theratoxin receptor transgene, and then wait for regeneration and see whether the newly formed cells are labeled or not. So this is what we did. And as you can see in this example, this is an islet after beta cell ablation. You can only see basically fluorescent cells that are alpha cells. And here you see one cell that is, in fact, an insulin-containing cell, but labeled with the reporter, meaning that this is a reprogrammed alpha cell. So with this type of experimental approach, what we have seen is this reprogramming sequence of alpha cells, whereby by destroying the beta cells, some kind of signal makes that about 2% or so of the adult cell population, alpha cell population, undergoes the expression of beta cell-specific transcription factors, such as PDX1, NK6.1, and these cells become bihormonal. They co-express glucagon and insulin before becoming fully reprogrammed alpha cells that make only insulin and express beta cell maturity markers. So the question that we are trying to address in my laboratory is whether this process is influenced by age or by gender, or whether other cells besides alpha cells can also undergo reprogramming into insulin production. We, of course, we're trying to determine the signal, the nature of the signals that make that some alpha cells start making insulin. We try to understand why only so few alpha cells seem to be prone to become insulin producers. And of course, we want to determine whether human alpha cells can also do the same. So by doing this beta cell-specific massive ablation at different times of a mouse lifetime, whether in adulthood or in very aged mice, or in juveniles before puberty, what we have been able to see is that the pancreas can reconstitute throughout life new insulin-producing cells via these cell-type interconversion events. So, to make it short, we see that this alpha cell plasticity is preserved also in very aged mice, which is nice, it's very good news from a therapeutic perspective, and also that in early life, before puberty, prepubescent mice have a very robust capacity of regeneration that leads to recovery of diabetes. And in this situation, the phenomenon, what we observe is that delta cells become insulin-producing cells. So what happens can be summarized in this slide. During development, pancreas development, we have this stage-specific fate allocation of cells from multipotent precursors. But what happens after beta cell injury postnatally is that we have this kind of spontaneous cell-type interconversions. In juvenile mice, we have this epimorphosis whereby delta cells de-differentiate en masse, they replicate, and they become bipotent, so that they give rise to new populations of delta cells and beta cells. In adult mice, what happens is the morphallaxis process, and it concerns not only alpha cells, but also delta cells. About 1 or 2% of the alpha cell population and 1 or 2% of the delta cell population in adult mice undergoes this direct conversion, so that they become hybrid, they become bihormonal, they co-express glucagon and insulin or somatostatin and insulin before becoming fully insulin expressors. Another set of experiments are trying to determine whether the mode of beta cell death triggers the plasticity in alpha cells, or whether this plasticity is observed only after a total loss of beta cells. These experiments were performed by Valentina, and she was using streptozotocin and aloxan. As you know, these are glucose analogs that enter beta cells through GLUT2, and they both result in beta cell death, whether by breaking DNA or by producing free radicals, reactive oxygen species. They trigger the death of beta cells by necrosis, and in this system, the efficiency of beta cell ablation is slightly lower than with diphtheria toxin. About 70 to 90% of the beta cells are destroyed with streptozotocin. Just to summarize, what she observed in these classical models of experimental diabetes is exactly the same. So a total beta cell loss is not a requirement for having this cell fate conversion of alpha cells and delta cells. And these things also happen independently of the mode of beta cell death, because in these animal models, beta cell death is by necrosis, whereby with the steatoxin is by apoptosis. So these are important pieces of information that I don't have the time to describe today in more detail. So we can speculate about human islet cell plasticity, since we know that in mice, in absence of autoimmunity and in absence of chronic metabolic alterations, we have seen there is spontaneous beta cell reconstitution by heterologous conversion. This means that regeneration by reprogramming, this plasticity, is something that happens naturally in the injured pancreas. So in the lab, Kenichiro Furuyama wants to determine whether human alpha cells can reprogram into insulin production too. So we have seen that in mice, we have this alpha and delta cell plasticity. The question about human islets is not whether in human patients, in human people with diabetes, we have these cell interconversion events. The question is rather to try to understand the pathways and factors that promote or facilitate this plasticity in order to try to induce this to occur in the pancreas. So there are plenty of evidences now in human diabetics that led us to think that perhaps there is some plasticity. In fact, this is an islet from a type 2 patient, and in this islet you can see these two cells that co-express glucagon and insulin. Here, there is another sample from a type 1 diabetic, and you see one cell, the very cell containing both somatostatin and insulin. So there are bihormonal cells at higher frequency in diabetics. And this makes us think that perhaps there is also this kind of plasticity in the human pancreas. There's also a set of interesting observations in children with pancreatic tumors or insulinomas that have to undergo this subtotal pancreatectomy. They become diabetic because of this pancreas removal. And in many medical reports, it's clear that most of these children become insulin independent with time, suggesting that indeed there is regeneration. And we like to think that perhaps this is through this adaptive islet non-beta cell conversion. So what Kenichiro is doing in the laboratory is to take human islets from non-diabetic donors in general, dissociate them with highly specific antibodies that are commercially available by flow cytometry, and very efficiently he can get pure alpha cell populations and pure beta cell populations. Sometimes he also gets pure PP cell populations. So with these cells purified, he can label them using adenoviruses that encodes for a reporter fluorescent protein. And in this example, for instance, after a week of transduction, he sees that indeed the large majority, 99% of these cells are in fact fluorescent labeled. They are YFP positive. So what he can do with that is plenty of experiments. He can reaggregate them to generate pseudo-islets, and then do in vitro analysis or transplant these pseudo-islets into mice. So human islet cells can be lineage traced in long-term studies. So with these pure islet cell populations, we can do plenty of things. We can reaggregate them and play with the number of cells, but also with the composition of these pseudo-islets. And today I will talk about some experiments which concern the use of monotypic beta cells, pseudo-islets, and monotypic alpha cell pseudo-islets. So first, as a proof of concept that this works well, I choose to talk about this experiment in which we have these isolated beta cells that, as you know well, they are not good at secreting insulin in response to glucose in isolation. So what Kenichiro did is a number of experiments, single-cell culture, reaggregation, or even reaggregation using these commercially available human vascular endothelial cells, mesenchymal stem cells. So in this situation, beta cells are very easily aggregated. Here you see in 5 days, this is a pseudo-islet of beta cells only. Beta cells are labeled fluorescently here. And when we use Hubeck cells, this reaggregation is even faster. You see in 1 day you have already these pseudo-islets formed. And interestingly, the mesenchymal cells always occupy the center of the aggregates and the beta cells remain at the periphery. But the interesting thing is that these aggregates of beta cells secrete insulin very nicely in response to glucose. So you see the levels of insulin and PDX expression in beta cells before aggregation and after aggregation, and you see the difference. And also in response to glucose, as I said, these aggregates of pure beta cells or beta cells with Hubeck cells secrete insulin in response to glucose. And importantly, this stimulation index is like in native islets before any dissociation. So monotypic beta cell only pseudo-islets are able to restore beta cell function. And this is an interesting observation. It's a confirmation of something that we already knew, which is that these heterotypic contacts between beta cells and non-beta cells through gap junctions or whatever are not required for normal beta cell function. So as you see, this process of adaptive change of cell identity of differentiated cells in response to stress or injury is an early discovery. It's a newly described mode of natural post-injury regeneration in mammals. So now what we have to do is to devise ambitious approaches of systems biology, of epigenetics, bioinformatics, to try to find a way to induce this plasticity. We have to develop tools, mainly by pharmacology, to force the cells to undergo this conversion. And we should exploit this plasticity to modeling the disease and perhaps think that we are maybe better at producing beta-like cells rather than beta cells, perfect beta cells. So I believe that the selective modulation of autoimmunity, the control or modulation of these chronic metabolic alterations may reveal in patients that the pancreas can in fact regrow new beta cells, as we have seen to occur in mice. And I believe, like this author in this review wrote at the end, that this approach, the capacity of controlling adaptive changes in cell fate in vivo, in order to promote the body's own cell therapy by pharmacology, is an area of increasing work in the future. I think that these experiments, still preliminary, but they already provide the necessary proof of principle for a novel therapeutic approach of fostering this naturally occurring beta cell regeneration process of adaptive cell identity change in islet endocrine non-beta cells inside in the body. So this is interesting also from a therapeutic perspective because reprogramming alpha cells into insulin production will necessarily encompass a reduction of glucagon expression, and this will be beneficial for diabetes because they will limit hyperglucagonemia and the hepatic glucose mobilization without any major metabolic effect due to this relative loss of alpha cells. So I want to conclude with this. I want to mention all the people that was involved in some different aspects of these experiments. Now, Simona, Luisa, Delphine, and Nicolas are gone. They are in other laboratories. They are very brilliant, very good scientists. This slide is just to thank the funding agencies and my ongoing collaborators. Particularly, we get islets from this integrated islet distribution program of the United States. And this is it. This is what I wanted to tell today from sunny Geneva. It's not always sunny, don't believe me. Thank you for your attention.